Genome shuffles, a new way to twist and shout about structural variants. Have you ever tried to understand the complex dance that our genes do? Well, researchers at the University of Washington are shaking things up in genetic research with their latest findings published in Science on January 31, 2025. Under the guidance of lead author Sudarshan Pinglay, this team of innovators has taken a monumental leap in our ability to study structural variants, SVs, in mammalian genomes. Picture a party where the dance floor of your DNA is getting an exciting new DJ, and that DJ is their newly developed technique, Genome-Shuffle-seq. So, what's the skinny on these structural variants? Think of them as gigantic rearrangements in the genetic code, the big movers and shakers, if you will. They include deletions, insertions, inversions, and even translocations that can significantly impact health and disease. However, pinpointing and understanding their roles has been like trying to find a needle in a haystack. Traditional methods have been limited, often producing only a few variants for study at a time. Enter Genome-Shuffle-seq. This ingenious technique allows scientists to generate and analyze thousands of SVs in one fell swoop. Using what they call shuffle cassettes, the team can create variations every which way and track their effects without the usual wheeling, dealing, and extensive cloning that makes genetics feel like a game of Monopoly that never ends. The result? More than 5,000 novel variants in just one go during test experiments on cultured mouse stem cells and human cancer cells. Talk about a serious upgrade from the old days of genetic analysis. Now, before you get up and start a conga line in celebration, it's important to note the reality check that comes with innovation. This research isn't without its hiccups. The structural variants initiated by one recombinase, CRE, seem to be as social as a tofu-eating hipster at a barbecue, eventually disappearing from cell populations due to toxicity. Meanwhile, its counterpart BXB1 was a much more stable guest, making its presence felt long after the party started. While these findings are promising, there are still some technical hurdles to leap. The cells often had a limited retention of the SVs generated by CRE, illustrating that keeping them around long enough for study can be a real test of patience. The hope is that using different techniques and possibly looking toward new technologies will crank up the endurance of these variants for future research. So what does all of this mean, exactly? Well, the implications of this research go far beyond the pages of a scientific journal. Genome-Shuffle-seq is poised to fundamentally change how we explore and manipulate genomes, potentially leading to insights into complex diseases, therapies for genetic disorders, and the very architecture of our DNA. Imagine a future where we can tailor medical treatments at a granular level by understanding how individual SVs contribute to conditions like cancer or heart disease. The research not only opens a pathway for developing finely tuned therapies, but might also pave the way towards the engineering of a minimal human genome, which could be revolutionary in fields like synthetic biology. So, whether you're a seasoned genetic researcher or just someone interested in how our bodies are built at the molecular level, Genome-Shuffle-seq is worth getting excited about. The potential for practical applications is huge, from enhancing our understanding of genetics to driving innovations in healthcare.